sure my computer doesn't fall asleep because it just keeps switching.
I will do that.
You just...
Okay, because you're... Hello. If everyone could please quickly take their seats. Good evening, and welcome to Cosyne 2022. I am so happy on behalf of all the organizers of this meeting, the board, my fellow General Chair, Surgeon Ostojic. We are so happy to host this first in-person meeting since 2020. 2020, wow, it's a long time ago. And honestly, if we weren't all already struggling with facial recognition that we do at every meeting we go to, I'm going to offer you a new challenge. We've all aged 2 years. The grad students are getting younger. Those that were grad students are postdocs now. Those that were postdocs, some are faculty. And we're all wearing masks. So good luck with the facial recognition this year. And so with that, I'd like to say I want to get this meeting started, and I'm super happy this year to introduce our program chairs. They have done an incredible job putting together this meeting. This has not been an easy undertaking, and they have weathered all the storms, and this is their chance now tonight to put on this incredible show that they've put together for you. So without further ado, I'd like to introduce Tim Vogels and Laura Busse, our two program chairs for Cosyne 2020.
Are you muted? I think you're muted.
Okay, also on my behalf and the entire organizing committee for Cosyne, I'm really happy to welcome all of you here. Like Anne-Marie was saying, Cosyne 2020 was probably the last meeting in person for many of us, and here we are again, the first in-person meeting, Cosyne 2022.
All right. Good evening. I'm really excited to be here. I figured we're going to talk about important questions, so we should put the most important question for experimentalists up right away. Where can I get those 5G chips? And do they work on rodents? I hope you're not going to find them here. It's not super likely that all of you are not going to go home with 5G, but I hope as few as possible. Also for theorists, we have an important question that's been going on Twitter. So I hope you're going to solve that one. All right. Here's another chore for Cosyne, a word cloud, completely useless, ugly. Maybe not completely useless.
From the word cloud, we picked topics that we thought were important for taking over the breadth and the depth of computational neuroscience. So this is what we chose as topics for our program. We built them into sessions.
As you can see. Most of them are actually relatively standard sessions, right? Learning, communication, memory. But there's a couple of cool ones. Empathy, immune systems, maybe neural space. More excitingly, maybe, we've assembled a crew of speakers that we're very excited about. I hope you're excited about them too. Here you see them in their most beautiful. Of course, it's not just these speakers. It's another 30 solicited talks. We didn't have photos for you, so apologies. And not just that. There's poster sessions to go to as well, and then there's a workshop to go to afterwards. So I think we're looking at a relatively exciting program.
Super. So COVID has and will mess with us. So we are really thankful for all your flexibility. This is flexibility on the side of the speakers who were able to last minute step in. There's flexibility in terms of talks that we have to shift or posters where we needed to go from one day to the other. So thank you very much for your flexibility. And I think probably some of these spontaneous changes will still need to happen during the conference.
This also extends to 3 speakers that had to drop out because of COVID. We're thinking of you. We're not naming you. But hopefully we'll see you next year.
Okay, so speaking of COVID, there are a few very important rules. So the first one, maybe the most important one, is if you're indoors here at the conference center, mask at all times, except when you're eating and drinking. So this is really important because it protects us all. Everybody that comes in here is had to show a proof of vaccination. And
put your mask on first and then help others.
So everybody who's in here had to show the proof of vaccination, but of course you can also develop COVID even if you're fully vaccinated. So we would ask to everybody to do voluntary self-tests regularly. You can get these at any Portuguese pharmacy. Test yourself. If you have any symptoms, if you don't feel well, please don't come. Please take a test and stay away from Cosyne.
Speaking of rules, there's also rules for Cosyne. One of them is phrased jokingly, play nice. The background is not so jokingly. We have a policy of conduct or a code of conduct. We would like you all to obey to it. Likewise, as if you had COVID, if you can't obey to it, please do stay home. Also, attend every talk and attend every poster, and there will be a test. That's actually made easy for you, even if you have COVID, because we're going to stream all talks. And this year, for the first time, we're also going to create something that's called audio briefs. So we asked poster presenters to create audio tracks for their posters. And you can pick up those audio tracks. Currently, I think around 100 people have uploaded audio tracks. Hopefully, they will present QR codes on their posters so that you can scan them and find your way to a website here that's provided by Worldwide Neuro. That features an audio file that you can click on and listen to the talk without having to necessarily engage with the presenter, which is always nice, right? There's also recommendations. So scroll down on that webpage and find the other most similar posters to the one that you're looking at.
Besides talks and posters, there's even more highlights. So I want to quickly mention the diversity lunch that takes place Saturday at 1 p.m., a panel discussion about the pandemic's effects on inequities that will take place Saturday at 4 p.m. And then we have a new way of engaging with this community. So there's a professional development panel and social Friday at 7 p.m. And I hope we will see many of you in all of these events.
Speaking of diversity, you might want to know how we all ended up here. First, maybe diversity of the program committee. We were tasked with increasing the diversity of the program committee from what was originally 1/3 women, the rest men. And that's an effort that's been going on over the years with members cycling out, new members cycling in. This year, we've reached 50-50 in men and women with a relatively good representation of topics. This, of course, says nothing about the representation of other minorities, so we're only looking here at male, female, gender ratios. But that's where we stand. So thank you to the PC members that are here and the ones that couldn't come, and also thank you to 215 reviewers that were recruited then by the PC members to do most of the work, which was looking at the abstracts and ranking them.
Okay, so let's see beyond the program committee who has applied to Cosyne, who is here. So this year we received, as you know from the emails, we received a total of 881 abstracts. You see a bit of a statistics on the gender ratio on the right side here. So we had 25% of first author female applications or abstracts submitted and 13% last author females. We also asked during the submission process if you identify yourself as a minority in computational and systems neuroscience, and here we got about 14% first authors who identified themselves as minority and 6% last authors. So I think these numbers are relatively constant compared to the year 2020. It is already a good ratio, but we also have some way to go, both with respect to gender as well as to minorities.
Thanks to the double-blind review process, male and female submissions, regardless of whether they're male or female last authors or male or female first authors, are indistinguishable from one another now in terms of their ranking. So we think that's a good thing. This is something that Sergeant and Anne-Marie introduced 2 years ago. And of course, I should say the reviewer assignment worked through an algorithm that is also used by Neuromatch. I hope you've all heard of Neuromatch. And that was devised by Tidipat, Daniel, and Konrad Kerding in Konrad Kerding's lab. And we've been using it for a few years, so thank you also at this point for letting us use this code for the foreseeable future, actually.
Super, so how do we go from these 881 abstracts that were submitted to an accepted abstract? So these 881 abstracts were sent out to the reviewers for their ranking. Each of these abstracts, or most of the abstracts, actually received 3 reviews. Tim and me then looked at the reviews and picked the top 390 abstracts based on the ranking from the reviewers. And we had some extra 90 abstracts that we looked at in more detail, and we were able to add to the 390 abstracts that are usually accepted for Cosyne. So we ended up with a total of 480 accepted abstracts, and want to congratulate all of you here who have one of these.
Maybe just to clarify, this was a luxury we had thanks to COVID, so that we could expect more than a few people to drop out. In fact, not that many people dropped out, but we also had more space here, so we could just handpick an extra 90 abstracts that we thought would add to the breadth and the diversity and multiple different facets of the meeting, and we enjoyed doing that. It was great. So we ended up with 480 accepted abstracts.
These 480 abstracts we looked at in more detail. We chose the top 100 of those based on the reviewers' ranks. We then looked at these in more detail and then handed over to our program committee 60 of those 100 to review again, to send comments and to judge, basically. And from these 60 that were ranked by our program committee again, we selected the 30 talks that ended up in the solicited talk section here, based on the ranking, of course, but also other aspects like the balance between computation and experiment, the topic where the first and last author came from, the model organism that they were studying, and other aspects like the gender and maybe minority aspects of the talks.
All right, those were 30 talks. We're looking forward to them. We actually had 0 dropouts, or maybe 1 dropout who couldn't make it, but we're looking very much forward to them. Of course, diversity also goes beyond choosing talks. That job was mostly done by other people here, but we have a strong diversity, equity, and inclusion committee that's headed by Gabrielle and Stefano, whose last name I can't pronounce, so I'm just going to pretend that I'm on a first name basis with them. And I also maybe wanted to draw special attention to a childcare grant program that Cosyne has been organizing for a few years now, and that has been particularly poorly done in terms of applications this year. So do tell your friends with children that we have this for the coming years, as well as other travel grants, as well as other access if you want to stay home.
So throughout the process, when we were trying to organize this meeting, we were often wondering what could we do better. So we had questions like, what is the correct or right rejection rate? What should be the breadth of topics? Are we missing out on anything in our description of what we want here to have here at Cosyne? Was this review process that everybody had to go through internally consistent?
Maybe another thing that we talked about was whether we should actually reject posters from labs that have multiple submissions. So we noticed that even though 75% or maybe more of the posters were submitted as a single submission from a given lab, some labs, and I'm not going to name any here, but we all have our suspicions, submitted up to 11 posters. And so that kind of breaks what's called the pillow rule at Cosyne. And it also, sadly, led to the fact that 244 out of 612 last authors didn't get a single submission in. And we were sad about that, because I think Cosyne is also an educational opportunity for grantees or of people that are accepted. So many of these, of course, are from those who only submitted a single poster. And you can see that even if we rejected every second poster from any given lab, we'd still not make up for the 244 that were rejected. But it's something to think about. And in fact, I think you probably all have some criticisms, some things that we did wrong. I'm sure we've got blind spots. We've discovered, for example, one blind spot when we chose session chairs. We only looked for gender balance, but didn't look for minority balance otherwise, because we were hectic and rushed and we made mistakes. So you'll find that our selection of session chairs is suboptimal. If you see anything else that you find suboptimal, do tell us. Do tell us offline. It's just nicer to get criticism, not in front of everyone else.
So with that, we're at the end of our opening section. We want to thank you for being here. We want to also thank our funders that make it possible to run Cosyne. This is the Boros Wellcome Fund, DeepMind, Gatsby, the Simons Foundation, and Reality Labs. As you heard, we are very happy that we have a great program committee, and we are thanking all the reviewers for the hard work they put to make this meeting possible.
And then we'd like to thank especially Leslie Weeks and Thomas. I'm going to try this one, Hramadka. They were really the wheels behind them in the background. They were the machine that created the infrastructure for all of this. They have been for years, and it's been a pleasure working with them. I would also like to make a shout out to Panos Potselos. He's been instrumental in getting the multimedia briefs off the ground. And I think many of you have probably encountered Panos' work over the last 2 years, because he's the sole driving force behind Worldwide Neuro. And so he's made all of our lockdown, all of the last 2 years that we've experienced, a little bit easier. So thank you, Panos. He's here, so if you see him, do embarrass him by telling him that you liked. So he's sitting over there, Worldwide Neuro. And then there's Carlos Stein and Grace Lindsay, who've been running very professionally video streaming, and who are running video streaming right now, and our social media accounts. So special shout out to them as well. Thank you. And with that, I think we'll end this one. Another Zoom joke for you. And without further ado, welcome to Cosyne 2022.
I'm going to try to speak through the podium mic here. Seems okay? Hi, everyone. Good evening. My name is Bing Brunton from the University of Washington, Seattle, and it is my distinct pleasure to be chair of the first session of the evening on the wonderful topic of brains and behavior. So after that, we should have all the questions solved. No need for other sessions. Our keynote speaker today is Professor Bob Datta, who is from Harvard Medical School, where he also received his MD and PhD degrees. We're really happy to have him here because his lab has done some of the most pioneering work on understanding the genetic and neural basis of natural behaviors. Animals doing whatever it is that they do in the wild, animals that have evolved to do the behaviors that they do. And his lab has especially made a lot of efforts to understand social behaviors, as well as behaviors that are of relevance to psychiatry. So one of the things that his lab is most well known for is the MoSeq analysis, this way of taking high-resolution video of mice behaving and then breaking it down to meaningful motifs and behavioral syllables. And using this language of behavior, his lab has done a lot of great work in trying to understand how it is that we can connect that language of behavior with things like neurocircuitry as well as psychopharmacology. So I know that I'm not alone in this audience who have been greatly inspired by his work over the years, not only because of the insights that we've gained from them, but also because of the tools that he has developed, because they allow us to answer questions, and well, maybe not answer, but ask questions that would have been really difficult to ask before in diverse animals. So before I hand over the podium to Bob, I'm going to say a word about asking questions. So this is a big room, and we've never run this session in this room before, and so there's going to be 2 people with microphones who are going to be running around handing microphones to people in the live audience who have questions at the end of the talk. So just be thinking about questions that you might want to ask. And we also have some questions coming in from the live streaming audience, and so this will be an experiment. You can do this experiment with us. We're going to try to figure out how it is that we can have some live questions as well as some questions that are from online. Okay. We especially strongly encourage those of you who are maybe newcomers, graduate students, postdocs, to be brave and ask questions. Okay. If you're sitting next to a friend who has a question, give them a nudge, have them raise their hand. I'll call on you. All right. Please join me in welcoming Bob Datta.
Everyone can hear me? We're good? Sounds like we're good. Okay, great. So first off, thanks, Bing, for that very kind and generous introduction. Thanks to the organizers, of course, for inviting me. It's a great honor and pleasure. And thanks to all of you for coming out in person. It's just so great to be at a meeting where we can kind of exchange scientific ideas in real life. I'll just give you a bit of a heads up. I plan to babble on for about an hour. The first 40 minutes or so are going to be stage setting and kind of review of stuff that we've published in this space. So those of you who are familiar with my work, take that as your cue to fall asleep now. But the last 20 minutes is all going to be kind of new, interesting, unpublished stuff. And so, wake up at about that point. So let's begin. So the behavior of animals in the natural world, where they're free to express whatever actions they choose, whatever order they want, of their own free will and accord and in their own time, is obviously remarkably complex and kind of delightfully varied, right, both within individuals and across individuals. And because of all of this, to us as human observers, it often appears to be rather inscrutable. But the ethologists argued that this kind of behavior, naturalistic, spontaneous behavior, is actually characterized by an enormous amount of structure. They reasoned that natural behavior is actually composed out of behavioral modules, repeatedly used stereotype motifs of action or movement that are placed somewhat probabilistically into sequences and also are organized hierarchically in time. And they argued that this architecture, this underlying architecture for behavior, is selected for by evolution and enables animals to both meaningfully interact with their particular ecological niche, but also to adapt to changes in context and contingency. Better? Oh, it's so much better. It's better for me. Okay. So they also argued, I think, a thing that all of us would agree to and is kind of quite intuitive, which is that if there is indeed a kind of underlying structure to behavior, ultimately it must find its origin somehow in the nervous system. But despite the fact that this sentence was written now nearly 60 years ago, it's arguably true that we still know very, very little about how the brain composes ongoing patterns of spontaneous behavior, the kind of behavior that most animals spend most of their lives expressing, including us as humans, the kind of behavior that happens in the absence of rigid task structure or some sort of predictable reward at the end of some explicit accomplishment or goal, the kind of behavior that occurs in the absence of physical, psychological, behavioral restraint. And so, there's this kind of central question that's really been haunting my lab for the last 5 to 10 years, right? How does the brain compose ongoing spontaneous behavior? We've been building tools whose goal is effectively to query this kind of ethological hypothesis that behavior might be characterized in some way as being modular. And our kind of first steps down this road actually came when a graduate student of mine named Alex Wolchko, who now runs a lab at Google Brain decided just basically for fun to replace the standard 2D cameras that we kind of were using for behavioral imaging with a 3D depth camera that he purchased from the Best Buy, which is just up the street from my lab. Here you can see one of these early depth images here. This is a mouse that we're imaging in 3D from a single axis of view using a depth camera. Heat mapped over this mouse at any given pixel, it's its height. And so when the mouse's head turns white, it means it's rearing up or sniffing. And currently in the lab, we're building all sorts of fancy machines where we image mice from multiple perspectives and can see the whole 3D carapace of the mouse and so on and so forth. But even from this relatively restricted view, right, just looking at the depth data from the top, I think it's quite obvious that that mice express really rich three-dimensional pose dynamics that are well captured by even this single depth camera. And the question is, given access to these kinds of data, how can we begin to make sense of the underlying structure of spontaneous mouse behavior? And insight came when Alex did a really simple thing. He decided just to dimensionally reduce these data. So this here, when I hit play, is going to be a movie of a mouse exploring the circular open field. We're going to extract the image of the mouse from the arena. We're going to align it so that its nose is to the right and so its tail is to the left. And Alex is just going to compress these data through the use, just for visualization purposes, of the random projections technique. And so this kind of stream is, these are the same data. And what you can see as this kind of compressed data plays out by eye is that it is very naturally striated. It seems to be kind of naturally and of its kind of, to be broken up into these little bits. And if we use fairly straightforward math to look at these bits, it seems like each one of these is 300, 400, 500 milliseconds long. And so that suggests that there might be a kind of underlying sub-second architecture that governs spontaneous mouse behavior, and that each one of these little bits might represent, in effect, one of these modules of behavior that the ethologists were talking about. And so to begin to explore that as an idea, Alex, along with Matt Johnson, who was a postdoc in my lab and who now himself runs a lab at Google Brain, developed a system, which you heard about, called motion sequencing, or MoSeq for short. And MoSeq is an unsupervised machine learning algorithm that, based on the inherent spatiotemporal structure of the behavioral data that we feed it, automatically identifies an optimal number of behavioral modules to describe or predict whatever kind of behavioral data we feed the algorithm. And I want to stress that MoSeq can take in as its input really any kind of time series behavioral data. So we've used it successfully to characterize mouse song. We've looked at human facial videos, accelerometer data. But for the purposes of this talk, everything I'm going to tell you about is this kind of 3D data of mice running around in these empty open fields. The MoSeq algorithm really is a generative model for mouse behavior, which instantiates this ethological hypothesis that behavior is built out of components that somewhat predictably tumble one after the other in time. It describes each behavioral module, which we also call a behavioral syllable, as a kind of autoregressive trajectory through high-dimensional pose space, with switching dynamics between these syllables imposed by a semi-Markov process that's focused on the sub-second time scale. And this whole model is fit using Bayesian non-parametric techniques. And what that means is that we can feed this model some data, and then we do some fitting, and what's spit out at us kind of automatically is a kind of optimal number and identity of these behavioral syllables. Now, I'm not going to actually go through any extensive validation of this method. We've published a number of papers using kind of MoSeq, and you can see all our controls and our thinking and our model comparisons in that context. But for those of you who are less familiar with how this technique works, I want to just briefly show you a few movies of these behavioral syllables to try to convince you that it's reasonable to think of mouse behavior as being at some level modular. So when I hit play, this is going to be a 3D movie of a mouse behaving in the circular open field. You can see its little head here, its ears, and you kind of see its tail down there. And the mouse is going to generate some behavior, and at some point in time, you'll see a dot come over the animal. When you see that dot, the mouse is expressing a MoSeq-identified behavioral syllable, which post hoc I have looked at and I have given a name to. I've decided to call it the move it along syllable because the animal is going to kind of scooch forward during the expression of the syllable. So here's the movie. There's some behavior. There's the dot. Animal scooches forward, and this is going to loop. And of course, the key question is, is this little motif of movement actually a stereotyped and repeatedly used element of behavior in the sense that the ethologist meant it? So here on the right are 30 separate instances of the same behavioral syllable, now drawn from 8 separate experiments and 8 separate mice. And here I'm going to overlay and time lock all of these movies. Because there are 30 separate examples, when I hit play, of course, all these mice are going to do all sorts of wild and different things. But when the dot goes over the animal, they'll all kind of coherently express this move it along syllable. As this movie loops, you'll notice that the duration of each one of these instances of this particular syllable varies a little. Sometimes it's long, sometimes it's short. And that is entirely by design. MoSeq identifies and fits a specific spatiotemporal distribution of variance around each syllable to acknowledge that no innate behavior is expressed in precisely the same way twice. In a typical open field behavior, MoSeq will identify 40, 50, 60 of these little behavioral syllables. That number, of course, varies because, as I mentioned, this is an unsupervised machine learning technique, so it depends on the genotype of the animals, the shape of the arena, whether there's affordances there, is there more than one animal, and all of those things. But in a typical OFA experiment, like I'm showing you here, with these crowd movies, we tend to discover 40 or 50 of these syllables. And often these syllables are things that we as humans could have a priori identified as reasonable behavioral motifs. Things like the scrunch make a lot of sense. But sometimes they're things that maybe we as humans wouldn't have noticed, but turn out to be kind of reliable and predictable elements of behavior. Like this down and dart behavior here, where animals are reared up and they rapidly drop to the floor and then dart forward. Again, because MoSeq is unsupervised, it also is capable of identifying relatively rare events that nevertheless are predictable and interesting. For example, you can see here on the lower right, when you put a mouse in a bucket for like 20 minutes, they get pretty bored and start to want to escape. And so this is a syllable associated with the animal trying to fling its body desperately outside of our little arena. Okay, so this is these are the set of parts. And of course, motion sequencing identifies the number and kind of identity of these parts and tells us how those parts are being expressed over time as a sequence during any given experiment. And we can synthesize all of that information together into one of these little behavioral state maps. So here, each node is a behavioral syllable. Its diameter reflects how often that syllable is used. And the thickness of these yellow directional arrows indicates the observed likelihood of transition from one syllable to the next over time. And I hope just by inspection you are convinced that the behavior of mice in my lab, just like the behavior of mice out in the natural world, is characterized by an enormous amount of structure. And the main question I want to talk with you about today is, how does the brain compose and create this kind of dynamical structure? Before I get to the good stuff, I want to spend just a few slides on a digression and aside. So we'll come back to that question. And I want to talk just for a moment about benchmarking. I'm sure many of the people in this community are aware that there's been this huge proliferation, this really salutary proliferation of machine vision and machine learning-based tools for characterizing behavior that promise to really reveal to us fundamental features of brain behavior relationships. And these tools fall roughly into two categories. There's a bunch of tools that are about measuring behavior and featurizing behavior, the sort of point tracking algorithms that I'm sure many in this audience have both developed and have used. Things like sleep and deep lab cut and dance fall into that category. And there's a whole other set of tools that kind of take featurized behavioral data as inputs. And then through either supervised or unsupervised machine learning methods, learn about behavioral categories. And of course, MoSeq falls into this kind of second class of analytical tool. And as these tools are developed, especially unsupervised tools for thinking about how behavior might be organized, I think there's been perhaps not enough conversation about the limits of all of these tools. Every one of these unsupervised tools comes baked in with a lot of assumptions that often are not transparent. And every method has its own strengths and weaknesses. And so I think it's important for us to, as a community, begin to think a little bit about how we're going to kind of make transparent and available to us an understanding of like what's good and what's bad about each of these algorithms that we're using so that we can understand our experiments. And so we've been thinking about how we might approach this problem. And I'll tell you one possible approach that we've published on. Our strategy was to develop a behavioral data set with an enormous amount of behavioral diversity. So what we did was we took 1,000 mice, and into each mouse we injected one of about, at this point, 25 different neuro or psychoactive drugs at one of 2 to 6 doses. So this is an enormous data set. We basically throw the mice in acutely after being injected into the open field, and we film each one of these mice for like 30 minutes, okay? So we have this like giant data set with a ton of behavioral diversity in it. And then for each mouse, we can build a behavioral fingerprint through some methodology that allows us to kind of compress down and describe the animal's overall pattern of action. So here on the right, you can see us doing this with motion sequencing for individual mice. This is a little barcode that just indicates how often each behavioral syllable was used. In this example experiment, we can also build a much more traditional behavioral fingerprint, the type of behavioral fingerprint that would be used by a kind of modern behavioral neuropsychopharmacologist trying to develop a drug. So this fingerprint would include information like what is the distribution of locations the mouse adopted in this experiment? How fast was it moving over time? What are its angular velocities? How often did it turn? And so we can create a very long list of these kinds of traditional descriptors, and we can make a kind of traditional behavioral fingerprint for each mouse. We can then submit these fingerprints to just a simple linear classifier and just ask, which description of behavior afforded us more insight in terms of predicting the drug and dose that each mouse was exposed to. What you're looking at here is, of course, a confusion matrix. On the y-axis are all the different drugs these mice were actually exposed to. On the x-axis are all the drugs that the linear classifier, based upon behavior alone, predicted that these mice were exposed to. Perfect performance in this kind of analysis is, of course, bright white along the diagonal. And what you can see is that if you amass a huge diversity of kind of traditional behavioral metrics to describe mouse behavior, it actually does a pretty poor job of allowing you to identify which drug the animal was on at any one of these experiments. But I don't know, circa 95% of the time, MoSeq tells you the drug, and maybe 90% of the time or so, MoSeq will tell you the drug and the dose. And what it tells you is that we have this kind of framework that allows us to quantitatively demonstrate that motion sequencing, which essentially is just seeking to describe behavior as being modular and isn't really trying to tell things apart at all, gives a description of behavior that's well suited to making fine behavioral distinctions. It hasn't escaped our attention that this large data set that we have that allows us to make this kind of comparison will allow us to essentially test the relative performance in this particular classification task of a whole slew of different possible ways of characterizing mouse behavior. It also allows us to inactivate or embellish MoSeq in various ways to ask whether, like little bells and whistles that we're adding, are helping or hurting. And I won't take you through any of those analyses. I'll just tell you two broad conclusions that we've drawn to date that are provisional from this kind of analysis. The first is that it seems in our analysis that a particular feature of MoSeq, which is that it's kind of governed by the semi-Markov process that seeks behavioral structure at the sub-second time scale. It seems that that really helps with MoSeq's ability to capture and describe mouse behavior in particular. The fact that MoSeq has a characteristic time scale and a single time scale at which it seeks to characterize behavior is, in a real sense, a weakness, because that focus on the sub-second time scale potentially and actually formally obscures at least some information about the hierarchical, longer time scale organization of behavior. Nevertheless, that kind of unitary focus on the sub-second time scale generates behavioral representations that are extremely performant in this kind of classification task. And in fact, it's this time series nature of MoSeq that allows it to outperform a number of different comparators. The other thing I want to say is really obvious, which is that we use these kind of depth cameras to see the animal in 3D, and so we have a real rich and continuous description of the mouse's overall three-dimensional pose. And that's different from what you typically get when you do point tracking, where you're kind of taking a number of points on the animal's surface, and you're kind of tracking those in 2 or 3D. And when we do comparisons, it's clear that 3D imaging helps, and the reason for that is quite obvious. The dimensionality of the data and the richness of the data, therefore, are correspondingly high, and that helps make fine distinctions in behavior. I don't think that's a surprising claim at all. But I think what's interesting, and I want to stress here, is that actually you can get a lot of the way there. You can do really, really well with point tracking. Actually, you can learn a lot about the animal's overall pattern of behavior just by looking at subsets of points on the animal's surface. And so we're really excited about that because I know that many of the people in this community use those kinds of data to characterize behavior in their experiments. And so along with a team that I'll describe at the end of folks that are working on MoSeq in the lab, we're building a kind of clever port that's going to allow us to characterize easily point tracking data via MoSeq. And we're hoping to release that publicly on short time scales as soon as we're confident that it works for everyone. But we're excited about that, and we think that's a really valuable future approach for the use of motion sequencing. Okay, diversion aside, let's get back to the problem at hand. How does the brain actually create the kind of underlying modular structure of behavior that seems to afford MoSeq the ability to make these fine distinctions? So our first real kind of almost mechanistic insight into this came from a very simple experiment that Alex did when he was still a graduate student. Half of my lab studies will faction. So one of our main interests is in just understanding how odors change patterns of behavior. So Alex did this very simple experiment. This is an odor box At each corner is something called an olfactometer, which is really just a valve that can introduce odors into specific quadrants here. Again, you're looking at 30 separate experiments, 30 separate mice. We're going to overlay all of these movies. In gray will be the average position of all the mice in this particular experiment. And in this first experiment, we're just going to blow air. And what you can see is normal mouse behavior. The mice circumnavigate this arena. They dart through the center. They love these little corners, right? This is normal mouse open field behavior. And this behavior is radically changed when, in this corner, we introduce a monomolecular odorant called trimethylthiazolene, or TMT, which is derived from fox feces. And when mice smell this, they freak out because they think they're going to get eaten by a fox. And you can see their whole behavioral pattern, their whole behavioral state changes. They do this approach and avoid behavior. They tend to freeze in these corners as far away from the odor as they possibly can. They generate escape behaviors. It's obvious their kind of complete behavioral state has been altered by the presence of this fictive fox. And the question is, from the perspective of motion sequencing, how did behavior actually change? Okay, so this is what we see when we build these state maps before and after exposing animals to the odor of the fox. Now, when we, for example, take an animal from one arena to the next, or maybe we give the animal an affordance, MoSeq is great at identifying brand new behavioral syllables. But here, if you guys are eagle-eyed and you can kind of match up nodes, what we find is that there are no new behavioral syllables that emerge as a consequence of exposing the animal to TMT. And so that means that even though the animal's overall behavioral state has changed and all of the behaviors it's generating are different, none of those new behaviors are the consequence fundamentally of new pose dynamics. And that means that the way that mice are composing these behaviors in response to TMT is via resequencing this existing set of behavioral syllables. And you can really see that clearly if you simply subtract off these two maps. And in red are all those temporal interconnections that are downregulated, and in blue are all those that are upregulated. And so every new action the animal took in this experiment as a consequence of confronting this fake predator is actually a trajectory through the blue part of this behavioral state space. And what's neat about motion sequencing is, of course, we can just look at movies. Now when the dot comes over the animal, you're not looking anymore at a single syllable. Instead, you're going to look at a series of 3 blue syllables. And when we inspect these, indeed, we find that the movies that emerge are precisely those kind of new behaviors that were elicited by TMT. And including this freezing behavior and this kind of motivated odor-guided avoidance behavior. And so this, I think, to me, says something pretty important, right? It says that, as has been demonstrated before, for example, in the worm and in the fly, that when an animal is confronted with a stimulus and has to generate a behavioral response, it doesn't necessarily generate that response through the kind of emission of fundamentally new pose dynamics. But instead, it's capable of composing new behaviors by resequencing the order in which a bunch of parts, pre-existing parts, are expressed in, right? And that got us thinking. This idea that behavior can be kind of dynamically kind of recomposed on the fly, that got us thinking about where in the brain might such a resequencing process actually occur. And that in turn, of course, got us thinking about corticobasal ganglia loops, and especially about the input nucleus to the basal ganglia, as shown here, the striatum. And that's, of course, because the striatum is well known for its defined role in motor control. And the many experiments that have suggested that, at least in some way, in collaboration with cortex, the striatum is involved in the process of helping animals to choose what to do next. And both of those processes, I think, are obviously potentially important to the composition of naturalistic behaviors. And we've chosen to focus on a specific subregion of the striatum, which isn't indicated here. It's called the dorsolateral striatum. And we look at this particular subregion for a couple of reasons. First, it receives really strong inputs from both motor cortex and proprioceptive cortex. So in principle, it's a good place to think about 3D pose dynamics being encoded or regulated in some way. And the other is that there's a body of work from many researchers, but I really want to highlight the work of Kent Berridge, that has suggested that the dorsolateral striatum in particular might be involved in kind of innate naturalistic behavioral sequences, and they've focused specifically on grooming. And so we saw those kind of previous data, and we wondered whether or not, essentially in general, maybe the dorsolateral striatum might encode behavioral syllables, behavioral grammar, or both. So to test this hypothesis, we, and here I mean Jeff Markowitz and Wynne Gillis. Jeff is a shared postdoc between my lab and Bernardo Sabatini's lab, and our collaborators, which include Bernardo and Scott Linderman, who's here in the audience today. who's at Stanford, have collectively rendered motion sequencing compatible with essentially every form of neural recording. And so now you can basically stick a probe, whatever probe you want, anywhere you want in the brain, let the animal run around, behave spontaneously, and begin to query brain-behavior relationships. And so let me show you one example of us doing this in dorsolateral striatum. So here is a 3D mouse running around in a bucket. Again, this mouse is being aligned like I showed you before. Its nose is to the right, its tail is to the left, but it's still running around freely. And you'll notice this mouse is wearing a hat out of which is coming a cable, and that's because we're performing mini-scope type imaging of populations of spiny projection neurons in the dorsolateral striatum, and we're assessing their activity by looking at calcium transients, as you can see here in this kind of movie. And so obviously, post hoc, we can take neural traces from these data and behavioral traces from these data and look at individual neural activity here and sync that up with the mouse's evolving expression of syllables over time. And I think you can see a little bit by inspection what ends up being the underlying truth of these data, which is that as animals express behavioral syllables, different ensembles of neurons in the dorsolateral striatum kind of switch on and off as animals flip between syllables from syllable to syllable. This turns out to be quite a systematic relationship. So here on the left is just us systematizing that. We can see this kind of trend in either photometry data or an aggregated miniscope data. On the x-axis here is time. Time 0 is the transition into any new syllable in our syllable set. And the y-axis is just activity. And you can see here there's this systematic temporal relationship between the firing of neurons in the dorsolateral striatum and the animal switching into every new behavioral syllable. It's like there's a neural rhythm in dorsolateral striatum that matches the behavioral rhythm at the sub-second time scale that's being identified by motion sequencing. Further, the ensembles that are being dialed in as animals begin to express each new syllable explicitly encode the identity of the particular syllable. You can see a little decoding analysis here. At the ensemble level, we can make accurate predictions about the ongoing three-dimensional movements of the mouse based upon this ensemble activity in dorsolateral striatum alone. Krause has actually seen very similar things. And so what this suggests is that the dorsolateral striatum contains rather explicit representations of behavioral syllables. We also wondered whether there was any information about the sequence context in which these individual behavioral syllables are being expressed. So there's lots of ways to think about that. We've chosen to think about it in a very simple way. So imagine you have a sequence of syllables, and we're going to focus here on syllable B, okay? So you might imagine that when you express syllable B, statistically, you tend to express a number of subsequent syllables, but those syllables occur with different frequencies. You might imagine that the most frequent choice is this syllable, syllable C. And so what that means is that the sequence A, B, C can be characterized as being highly predictable or deterministic or having low entropy, right? In contrast, every once in a while, at some low rate, you might imagine that this very same syllable, syllable B, is followed by another syllable, syllable D. And thus, the sequence A, B, D, we might consider as being more probabilistic or more entropic or more random or less predictable, okay? So we can just break up the instances of syllable B into those episodes in which B is participating in a more predictable sequence and compare it to what we see when B is participating in a less predictable sequence. And that's kind of shown here. I think this is photometry data. We see the same thing in endoscopics data. But across all syllables, we see the same trend, which is when a given syllable is participating in a less likely, kind of more random sequence, the neural activity associated with that particular syllable is higher. And conversely, when that syllable is much more predictable, kind of a ho-hum sort of sequence, the neural activity associated with that syllable is lower. And so it's clear that the dorsolateral striatum both encodes the identity of ongoing behavioral syllables and contains within it information relevant to the contextual sequencing of individual behavioral syllables, which led us to ask, is the dorsolateral striatum required in any way for either the implementation and execution of individual behavioral syllables, the assembly of these syllables into sequences, both, neither? And so we did a pretty simple experiment where we just bilaterally lesioned the dorsolateral striatum with NMDA. We then waited a couple weeks for the mice to recover, and then looked at them via motion sequencing. So here's a couple of movies. This is the sham control, and this is the lesion. And what you can see is that the behavior of this lesion mouse by eye looks pretty good. It looks pretty normal. And when we do motion sequencing, the actual syllables are identical between these two conditions. And when we do careful kinematic analysis within the resolution of our assays, it looks like the expression of each of these individual syllables in the lesion case is normal. And that, I think, pretty clearly suggests that the dorsolateral striatum, despite containing explicit codes for the identity of each syllable, actually isn't involved in the implementation of the execution of each syllable. In contrast, we see dramatic changes in the order in which these syllables are expressed during open field behavior. So here we're looking at two behavioral state maps. Here we're going to push all the nodes, all the syllables out to the outside, so we can really highlight the interconnections between syllables over time. So here you can see what I've shown you now many times before, which is that it's clear that there's a lot of structure to spontaneous behavior, that there are somehow rules that govern the order in which different syllables are allowed to be expressed. And these rules are essentially dissolved. Behavior becomes much more random if we get rid of the dorsolateral striatum. And that suggests that the dorsolateral striatum is playing some important role during naturalistic behavior in controlling the sequencing of individual behavioral elements. Now, there's one kind of, I don't know, bit of funny business here, right, which is that I just showed you a movie of this lesion mouse, and it looked pretty normal. And so I think it's reasonable to ask whether there's any consequence of actually randomizing the order in which these behavioral syllables occur. And so to do so, we turn to an experiment that I've already described to you. I'll remind you that when we give an animal TMT, this fox odor, they generate these robust avoidance behaviors, and they do so by resequencing their behavioral elements. And so we just asked, is a mouse with a lesion in the dorsolateral striatum, which is clearly, and such a mouse is clearly capable of smelling TMT, actually capable of generating the appropriate behavioral response? On the left is the sham lesion control animals. You'll see it approach this kind of TMT spot here and run away. Those of you who study non-human primates will be delighted to know that mice are really stupid. And so this mouse is going to go and reapproach this port and smell it again and run away. Obviously, it's a bad strategy in the real world. But this lesion mouse treats this TMT port as if it's not even there, despite the fact that we know that this animal is capable of smelling and detecting the trimethylthiazoline. Okay, so what is this? What have I told you thus far? I've told you that the spontaneous behavior of mice has this kind of structure. It seems like the dorsolateral striatum is playing an important role in encoding various aspects of this structure, including the identity of ongoing behavioral syllables and the kind of order in which they occur. And I've shown you this interesting experiment in which, if we lesion the dorsolateral striatum, the mouse suddenly kind of randomizes its behavior and more importantly is incapable of switching into an appropriate behavioral state that requires resequencing of behavioral elements. And all of this taken together is consistent with the broad idea that cortico-basal ganglia loops, acting probably through at least in part the dorsolateral striatum, play an important role in governing this kind of underlying structure to behavior. But this description, which up until now has been published work, leaves a number of, I think, very basic kind of zeroth order questions unanswered. For example, why are some syllables used more often than others? Why do some syllables tend to be expressed as part of highly deterministic sequences where they're embedded in sequences that go A to B to C, whereas other syllables, either on a given instance or on average, tend to be kind of embedded in much more probabilistic sequences? Where does all that come from? So a team of folks in the lab have been thinking about this, including Jeff Markowitz, who again is a joint postdoc between me and Bernardo Sabatini, and Wynne Gillis and Maya Jay, who are both graduate students in my lab, who are really awesome. Maya's here. Hi, Maya. And together with our collaborators, which again in this case include Bernardo Sabatini and Scott Linderman, but now we're adding the expertise of Nao Uchida. And collectively, we've been thinking about a potential role in the structuring of naturalistic behavior for the neuromodulatory inputs that impinge upon the dorsolateral striatum, which of course are dopaminergic. And in the case of the dorsolateral striatum, most of these inputs arise from the substantia nigra pars compacta. There are decades of work showing that dopamine acting in the striatum can both rewrite the architecture of electrical activity in the striatum, and of course dopamine plays a critical role in mediating and fostering corticostriatal plasticity, which is thought to be critical for enabling dopamine to play the role that it does as a kind of teaching signal, which I'll mention in a moment. It turns out that actually we know very little about what dopamine's doing during spontaneous behavior. In humans who have Parkinson's disease, obviously they have less dopamine, and they have difficulty with spontaneous movement initiation, with the invigoration and expression of movements, and with various forms of behavioral sequencing. And those have inspired mouse experiments, rodent experiments, to begin to explore potential relationships between dopamine and movement per se. And there are not that many results, but they fall broadly into two camps. So the first is exemplified by work from Dan Dombeck and Rui Costa and some others that suggests that when animals are stopped and then they initiate a new movement, that's associated with a little flicker of dopamine. And there's some evidence to suggest that these kind of initiation-associated dopamine transients are actually causal for the movement. Okay? That's one idea, that dopamine transients during spontaneous behavior kind of get you moving. There's another idea, a non-mutually exclusive set of ideas that are exemplified by work, but not limited to work, by Henry In's lab, in which maybe dopamine varies continuously, and the continuous variation in dopamine actually specifies the moment-to-moment ongoing kinematics of the animal as the animal kind of moves around in the natural world. That's the other idea. And again, I would say that there haven't actually been that many recordings, and so this is still an understudied topic where we don't know very much. In contrast, of course, there have been decades of work thinking about dopamine's role as essentially a teaching signal and kind of articulating a possible role for dopamine in sculpting behavior to enable animals to maximize their experience of reward in the future. Right? This is how we traditionally think about dopamine. But it's very difficult to think about such a role for dopamine during spontaneous behavior, especially in the context of the experiments that we're doing in my lab. We take an animal and we put it in a bucket with no sensory cues, no task structure, in the dark, right? And we just let it run around. And there's no rewards there, obviously, and no explicit goals. So it's not really obvious how this would play a role in shaping behavior. I'll say that. And it's also, given what we know about behavior, unclear how dopamine might kind of broadly relate to the ongoing movements of the animal, although there's some suggestions that it might. So we were wondering, whether through the lens of motion sequencing we might be able to identify any kind of meaningful relationship between dopamine and spontaneous behavior. And so we've begun to record dopamine transients here in dorsolateral striatum through the use of the dopamine reporter molecule D-light, which we express in spiny projection neurons in dorsolateral striatum itself. And so here we're going to perform photometry in this configuration, and you can see now behavior on the top, and these D-light fluctuations, these transients, here on the bottom. And the first thing you should all notice is there's a ton of these dopamine transients, many, many more, I would say, than I expected naively. And there are many more than can be kind of on the surface explained by movement initiation alone. We have done a lot of work trying to explain these transients in terms of ongoing movement kinematics, to think about whether these transients can somehow predict the ongoing actual movements of the animal. And to be honest, I don't want to harp on a negative result, but we haven't really been able to find any strong correlations, except for one thing, which seems a little counterintuitive, but I think is super interesting, which actually had been seen by Josh Dudman and a couple others in the past, which is that at very short time scales, there seems to be a negative correlation between the number of dopamine flickers you experience and the animal's overall velocity. More dopamine means at these short time scales, meaning you slow down. What's interesting is that this relationship actually inverts depending on the time scale at which we bin the data. So at longer time scales, at time scales of 30 seconds to a minute, you see this relationship inverts, and now we have the very comfortable relationship in which more dopamine means more movement. I think an idea that is kind of pervasive in the field. What about movement initiations explicitly? So here on the x-axis, again, is time. Time 0 is the moment at which the animal stops being still and initiates new movement. And you can indeed, as has been reported in the past, see these little flickers of dopamine that are associated with the animal initiating a new movement. But what's interesting is that if we don't consider just new movement initiation, but instead consider all movement transitions, that is, the transitions between all behavioral syllables, we now see much more profound modulation of dopamine in association with these kinds of behavioral transitions. These are the same y-axes. And it looks like on the surface, in this grand average, like if we're just telling an anthropocentric story, that as one syllable ends, kind of dopamine dips and exhausts itself, and then as animals initiate a new behavioral syllable, dopamine kind of spikes up, and that might invigorate or propel forward in some way the expression of the next behavioral syllable. That's a just so story, but it's a good way to think about it. This is what we see in the grand average, okay? Is this what we see for every behavioral syllable? So that answer is no. Here on the y-axis are just arrayed all the 40 different behavioral syllables that we're going to look at in this experiment. On the x-axis is time, where time 0 marks the moment at which you enter into a given behavioral syllable. And you can see that each individual behavioral syllable here is actually associated with a characteristic dopamine amplitude and waveform. This kind of top syllable over here, I believe, is a rear, and it's characterized by an early rise in dopamine, followed by this kind of fast decline. And in contrast, we have kind of, I think this is a pause down here, which starts out low and kind of slowly rises during the expression of the syllable. You can see here that, on average, each syllable seems to be associated with a kind of characteristic dopamine amplitude and waveform. I'll remind you all that I told you that when we did these recordings of spiny projection neurons themselves, the recipients of this dopamine, in the dorsolateral striatum, we could use that information to correctly decode or identify which syllable was expressed at any given moment in time. Here, it looks like, on average, the kind of waveforms in dopamine that are associated with each of these syllables is different. And so one might imagine that I should be able to use these dopamine waveforms to accurately predict which syllable is being expressed at any given moment in time. But it turns out that's wrong. And it's wrong because this average dopamine transient actually kind of hides, buried within it, a huge diversity of dopamine transients associated with this very same syllable. So indeed, while this rear is often associated with this large peak in dopamine, in many, many instances, it's actually associated with dopamine going in the opposite direction. Conversely, this kind of pause down here, although it's often associated with a kind of downward going deflection, it often can be associated with an upward going flexion. And I'll mention that this observation, that there's a diversity of dopamine transients associated with each syllable, is consistent with my previous claim that we don't see any correlations between these dopamine transients and movement kinematics. Because all 4 of these syllable instances, morphologically, kinematically, are basically identical, and yet they're all associated with different shapes and amplitudes of dopamine being associated with them, right? So that's consistent with my claim. So now we have kind of a problem. The problem is we have a clear temporal relationship between dopamine flickering, right, and behavioral switching as identified by MoSeq, right? Every time you express a new syllable, you get a systematic change in dopamine. But those changes don't tell us anything about the identity of the actual behavioral syllable that's being expressed. So what's going on here? One concern we had that I think is a reasonable concern is that maybe this is all just noise, that we're like foveating on this like really low level kind of fluctuation in dopamine that honestly doesn't mean a whole lot. And so we thought about how we might calibrate ourselves here. And so we decided that the way we were going to do this was to give the mouse a pleasant surprise. So we threw into the arena, just randomly, a bunch of delicious, and I know they're delicious because I've eaten them, chocolate chips, right? And then the mouse was going to kind of stumble into these chocolate chips, and somehow it innately knows that they're delicious, and begins to consume them and enjoy the deliciousness, which we know is rewarding for these mice, and in different contexts can actually be used to shape behavior. And then we can measure the dopamine transients that are associated with the mouse consuming this surprising and rewarding and delicious little treat. And when we do that, we see that the amplitude of the food reward-related dopamine transient that we observe in dorsolateral striatum is of approximately the same amplitude. It's higher, but it's not so much higher. It's of approximately the same amplitude as the dopamine fluctuations that we observe when the animal is just spontaneously running around in the absence of any reward or task structure. And that suggests something super interesting, right? It suggests the possibility that maybe dopamine in the moment is not commanding movement, but instead maybe it's shaping behavior in the future for syllables that it's associated with. And that suggests a very, very simple idea, right? Maybe if this idea is true, that dopamine is influencing future behavior based upon its association with individual syllables, maybe those syllables that I showed you that are associated with the most dopamine are precisely those syllables that are expressed the most in any given experiment. And that turns out to be true. So here you can see the correlation. Right here on the x-axis is the dopamine signal, on the y-axis is how often the syllable is used. And both within a given experiment and actually across experiments, we see this nice correlation in which those syllables that tend to be associated with the most dopamine are precisely those that are used the most, and conversely, those that are associated with little dopamine tend to be used the least. And the most interesting thing and the critical thing is that this relationship holds true within an experiment itself, as is shown here by this plot. So if the animal is kind of expressing behavior, and in a given instance of a given syllable, there's a large dopamine transient associated with that syllable, over a time course of about 3, 4, 5 minutes, the animal will express that specific syllable more over time. And conversely, if that particular syllable happens to be associated with low dopamine, the animal will express that particular syllable less over time. And I'll say that in the analysis that we've done, the association of dopamine with the syllable is quite temporally specific, and it doesn't really bleed over into the future or the past. It seems somehow associated with the actual syllable that's coincident with the dopamine transient. Okay, so it's clear that dopamine has this kind of interesting potential relationship with behavior in which it seems to be playing some sort of short-term reinforcement-like role, at least in principle here. I'll also just remind everyone that dopamine in kind of more structured experiments focused on goals and rewards and tasks has been shown to influence the behavioral choices that animals make, both in the moment and in the future. And so we just wondered whether or not syllable-associated dopamine also somehow regulated the sequence or the ordering with which syllables were unfolding over time. And so it turns out that those syllables, as shown here, that are associated with the most dopamine on average in a given experiment or between experiments, those dopamine-associated syllables tend to be embedded in the most variable behavioral sequences. And conversely, syllables that tend to be associated with little dopamine tend to be expressed kind of in a more ho-hum, predictable A to B to C sort of way. And again, this relationship holds true on an instance-by-instance basis. So in a given instance of a behavioral syllable, if it happens to be associated with a bunch of dopamine, the very next syllable it chooses to express tends to be a more random, less predictable syllable. And if that same syllable it happens to be associated with a low dopamine transient, in that particular instance, the next thing the animal does tends to be predictable. And this tendency to do more random things and associated with dopamine lasts 2 to 3 seconds. So I've talked to you about 3 correlations with dopamine, right? 1 is velocity, 2 is syllable usage over timescales of minutes, and the 3rd is behavioral variability on timescales of seconds. We have built a wealth of decoding and encoding models that predict these 3 behavioral features from dopamine or predict dopamine transients and fluctuations over time based upon these 3 behavioral characteristics. And what those experiments have told us is that these correlations that I'm describing to you here hold up nicely in these encoding and decoding models. And furthermore, it seems like all 3 of these behavioral features contribute to accurately making predictions about what dopamine looks like as animals generate spontaneous behavior. In the end, those are all correlations. We really wanted to do a causal experiment. We wanted to reach in and manipulate dopamine specifically during one of these behavioral syllables to ask what that manipulation would do to the animal's use of the syllable, its ongoing velocity, its sequence variability, what have you. And so we have rendered, through the magic of deep learning, motion sequencing closed loop. And so now we can look down at a mouse that's running around in an open field spontaneously, and with about a 50 millisecond or so latency, I can tell you which behavioral syllable out of the 40 it's expressing at any given moment in time. And we can use that to kick off an optogenetic trigger, which then allows us to make manipulations in any cell type or neural circuit we want. And that ultimately is going to allow us to falsify theories we have about the involvement of various kind of neural circuit stations in the composition of naturalistic behavior. And here, of course, our concern is with dopamine. And so the way that we're going to manipulate dopamine is by specifically stimulating dopaminergic axons that arise from midbrain dopaminergic neurons, specifically here within the dorsolateral striatum itself. So we're making a very spatially focal manipulation of dopamine just in this particular region that we've been recording in. Furthermore, we calibrate this experiment very carefully such that the light-evoked amount of dopamine that we see as reported to us by D-light roughly matches the transient size that we're observing as animals just spontaneously behave. And the consequence of this is that we're stimulating mice with 200 milliseconds of light at very low laser powers. This is a much, much more subtle optogenetic intervention than the vast majority of experiments in the reported literature that are trying to link dopamine to any aspect of behavior. And when we did this experiment, given its subtlety, we had every reason to expect that we would see no behavioral effect whatsoever. We're barely tapping on dopamine here at all. And indeed, when we look at movement kinematics, we see nothing. So whether we look at syllable-associated movement kinematics or movement in general, when we give these little flickers of dopamine, we add them to particular syllables, basically the animal acts from a movement perspective like they haven't seen any dopamine at all. And just to center our own understanding of this experiment, if we do what most people do, which is to not give 200 milliseconds of light, but instead to give 2 seconds of light, now we see all the expected and previously reported changes in behavior that are associated with high levels of dopamine, like an increase in the animal's velocity and turning behavior towards one side. And what this suggests to us is that perhaps dopamine, when it's fluctuating as animals generate spontaneous behavior, the amplitude of those fluctuations is not high enough to actually command movement on a moment-to-moment basis, at least within dorsolateral striatum. But we see something very different when we think about the more structural elements of behavior. So here now we're going to target a given behavioral syllable. This is just an example. We've done this to like 9 of the behavioral syllables thus far. Here are a couple baseline sessions. The targeted syllable is going to be in green here. This is just us looking at kind of control behavior. Now we're going to throw the mouse in the bucket and specifically pair a given behavioral syllable. I think this example is actually a rear, one of 40 behavioral syllables that's being expressed with a little bit of tickling of optogenetic dopamine. And what we see is that within 3 or 4 stimulations, so within the first couple minutes, this animal recognizes that there's some relationship between its expression of this kind of egocentric unit of behavior and this exogenous tiny little flicker of dopamine that we're adding to the system. And the animal immediately begins to upregulate its usage of that behavioral syllable. You can do this over 2 sessions. It happens in the first session. It happens in the second session. And we know that there's learning going on here because if you look at the latency with which the animals initiate that targeted behavioral syllable, on the second day, it's much quicker than it happens on the first day. And if we turn the light off, the animals kind of continue to overexpress the targeted associated behavioral syllable. And this is consistent with, what I just kind of implied in the endogenous data that perhaps, dopamine is sufficient to reinforce the expression of kind of associated syllables. And I want to pause for a moment and just say that, I think there's something pretty unusual going on here in this experiment that's worth at least noting. Typically we think of dopamine as being associated with rewards or sensory cues. And here again, the mouse lacks all of those things, right? There are no sensory cues telling it when the light's turning on. The only thing that it knows, right, is that it probably has access to some motor efferents, and it has access to proprioception and kind of information about its own body movements as it runs around by itself in the dark. And yet, based on that alone, it's capable of knowing what it's doing with its own body and reinforcing and upregulating, the targeted syllable, which I think is, pretty interesting. Okay, so, these behavioral syllables, as I've mentioned now many times, occur in sequences and in an order. So on those trials in which we add a little extra dopamine, what happens to behavioral variability in terms of the ordering of behavioral syllables? Well, as I told you in the endogenous data, we see the same exact result here, which is on those trials in which we add a little exogenous dopamine, the next behavioral choice the animal makes tends to be a much more random one rather than a much more predictable one. And again, this kind of injection of behavioral variability seems to last a couple seconds. Okay? And so taken together, these results kind of suggest that dopamine, at least in dorsolateral striatum, might not be commanding movement on a moment-to-moment basis, but instead might be influencing movement more indirectly by controlling the overall architecture of behavior, as shown here through controlling sequence variability, and as I showed you previously, through controlling the usage over timescales of minutes of individual syllables that are associated with dopamine. And that suggests a kind of interesting possibility where these kind of continuous looking fluctuations in dopamine that we observe during spontaneous behavior might be acting as a kind of ongoing teaching signal that shapes the architecture of the animal's behavior during spontaneous behavior, as it evolves over time. And so to kind of more formally explore that idea, we've built a simple reinforcement learning model. Here we have a reinforcement learning agent where the state the agent's in is the equivalent of the syllable the animal's expressing at any given moment in time. The action the animal generates is its choice of next syllable. And we've substituted in this simple reinforcement learning model the traditional reward signals for the dopamine that's associated with the syllable that the animal enters into next. And we're just going to use kind of generic off-the-shelf Q-learning rules to update a Q-table, or in this case a transition matrix. We're going to initialize this transition matrix uniformly. And this kind of says that at start, this reinforcement learning agent will kind of uniformly transition from any syllable into any other syllable. You notice 2 things. One is there are only 10 syllables here. That's because we're a little bit data limited. The other thing is that there's some black boxes here. That's just because those syllables aren't interconnected in time in the empirical data. But we can just kind of use Q-learning rules to train up this reinforcement learning agent, again, through standard techniques based upon observed behavioral data, right, and observed dopamine transients. And when we do that, we learn this transition matrix over here. And when we compare this learned transition matrix to the actual transition matrix that governs the animal's behavioral choices during behavior, right, in held-out data, we see a remarkable concordance between this learned transition matrix and what's observed. And that does suggest 2 things, right? It suggests first that dopamine is probably shaping, at least through this lens, the animal's architecture of its spontaneous behavior. And second, it suggests, doesn't prove it, it suggests that perhaps animals are interpreting these fluctuations in dopamine that it's experiencing during spontaneous behavior as a reward-like signal. I'll just mention one last thing here, which is that, I told you that dopamine has these 2 roles we think it's playing. We think it's kind of reinforcing, and we think that it's injecting variability. And if we embellish our model by including a little bit of this kind of syllable-attached variability over a couple seconds, we actually do slightly better at kind of matching the empirically observed transition matrices, consistent with dopamine playing both of these roles, as we have asserted causally and in the past through the use of the endogenous manipulation. The last data I want to show you relate to the question of how specific this is. Jonathan Whitlock has recently posted a beautiful paper, I think, on bioRxiv, describing the encoding of kind of syllable-like codes throughout the brain. And one might imagine that, like, the fact that we see the effects that we see in dorsolateral striatum is kind of generic, and we'd see these kinds of results anywhere in the brain. And so we wondered whether that was true. So we repeated basically all the experiments I showed you in dorsolateral striatum, in dorsomedial striatum. If we look at what's going on in dorsomedial striatum in terms of dopamine fluctuations, indeed we see these little flickers associated with transitions into behavioral syllables, but I will say that the amplitude of these flickers is a little lower than what we see in dorsolateral striatum. And we see at short time scales this correlation between dopamine and velocity that I described to you before. There's this kind of negative correlation. But we don't see any evidence in the endogenous data of a relationship between dopamine and future syllable use or dopamine and future behavioral variability. And indeed, when we go and do the causal experiment, and we stick our optogenic stimulation fiber not in dorsolateral, but in dorsomedial striatum, now we are unable to reinforce syllables that are associated with the optogenic dopamine. And this suggests that there actually is something special about dorsolateral striatum and kind of highlight its potential role during the composition of natural behavior. Okay, so just to conclude, I've described to you this system that we've developed called motion sequencing, which attempts to test this ethological hypothesis that behavior is modular. And so it identifies the putative modules of behavior or behavioral syllables for mouse behavior in the lab. The identification of these modules allows us to unveil this kind of structure, which you see here. I've told you that in dorsolateral striatum, there are neural correlates, both for syllables and grammar, and that it seems like dorsolateral striatum is important for essentially imposing order on this transition structure. And I've also told you a little bit of unpublished data about dopamine, how it fluctuates much more than I would expect. There are these pervasive dopamine fluctuations associated with spontaneous behavior, again, that's occurring in the absence of task structure or a reward. Right? And these dopamine fluctuations don't seem to be meaningfully changing the animal's ongoing behavioral kinematics, at least in dorsolateral striatum, but instead seem to be indirectly shaping the architecture of behavior by reinforcing the use of associated behavioral syllables and by injecting a little bit of variability into the expression of animals' ongoing behavioral sequences. I think one of the interesting things here is that when we think about animals making choices, we typically think about those problems from the perspective of much lower dimensional tasks where animals have a kind of explicit goal or are trying to maximize reward, right? And it's difficult to think about how circuits that are involved in those processes might relate to the inscrutable madness that seems to occur when you just let animals run around and do whatever they want. And what I think I've shown you here is that even in this very high dimensional case, in the absence of task structure, in the absence of reward, in the absence of any explicit goal, actually the very same circuits and some of the same computations might come into play in affording these kinds of behavior structure, as has been shown to afford kind of behavior structure in much more restricted tasks. And I think that's beautiful, that there's the potential for some sort of unified explanation for what dopamine does across these two circumstances that will allow us, those of us who are interested in spontaneous behavior, to have a real dialogue with those who are studying more reductionist preparations in the laboratory. Of course, the key unanswered question here is where are these dopamine fluctuations actually coming from, right? One model in extremis is that these fluctuations are just noise, right? And that you might imagine that animals are confused and just thinking that these fluctuations are some reward or error-like signal and shaping their behavior in response. We think that's not a very good idea because the behavior of spontaneous behavior has characteristic dynamics that are stereotyped across individual animals, suggesting that there's some rules that govern how animals explore little arenas and generate spontaneous behavior. So instead we think, of course, that animals are actually trying to achieve some sort of implicit goal as they explore an arena, and that these dopamine fluctuations reflect computations that are in support of this implicit goal. We don't know what the implicit goal is. We're thinking hard about what it might be. I think there are many possible ideas about what it could be, but we're excited to explore the possibility that the animal has a goal that we don't know about, and that the dopamine fluctuations we observe might be related to that implicit goal. Finally, I got tenure like 4 months ago, and so I'm allowed, given that this is a keynote lecture, to end my talk with some rank speculation that's almost certainly incorrect. I hope you'll all indulge me in that, because I want to have some fun. In kind of the behavioral neuroethology community, there are robust and important debates about whether it's best to describe behavior as being discrete, continuous, or both. And I think all of us know intuitively, right, that it has to at least at some level be both, because when you describe behavior instantaneously, that those descriptions are almost always best thought of as continuous. And of course, all of us fall asleep and wake up, and those are discrete states. And so the real question is, between these two timescales, between days and milliseconds, to what extent is behavior best explained as being continuous, discrete, or some combination of the two? And as we learn more about circuits that control behaviors, right, as we think about the hindbrain and the spinal cord and the hypothalamus and the amygdala, I think it's pretty intuitive and clear that the behavior at many timescales is going to have both discrete and continuous characteristics. For us, I think it's an important question to ask whether it's reasonable to think of behavior at the sub-second timescale as actually in some meaningful way being discrete. And I've told you now that when we look in the brain, which I think is a good way, to be honest, of adjudicating between models for behavior, I think thinking about neural correlations might be the best way to do that. We can argue about that later. But when we look in the brain, we see a neural rhythm, both in dopamine and in dorsolateral striatum, that seems to correspond with the animal switching from one behavioral syllable to the next. Consistent with the idea that it might be reasonable to think of behavior as being discrete. But I think what's really interesting is the optogenetic experiments that I've described to you. So during these experiments, there's a 50 millisecond offset before, after syllables begin, and then we shine the light for 200 milliseconds. Practically what that means is that our optogenetic transient, in many, many cases, is shorter than the duration of the syllable itself. If behavior were continuous, truly, you might expect that these kind of little flickers of dopamine might break syllables up into parts, or prompt switching into the next behavioral syllable, or somehow alter the morphology of associated syllables, given that the dopamine covers only a subset of the time that the syllable is being expressed. But instead, through every lens we can look at, reinforced syllables look identical to their unreinforced counterparts. Their duration distribution seems exactly the same. And the only thing that seems to be changing, their morphology is exactly the same too, the only thing that seems to be changing is kind of the overall average rate at which they're expressed within a given experiment. And that superficially suggests that maybe these behavioral syllables are actually kind of units that the brain can usefully use to organize action. And that has gotten my lab shifting in our thinking from thinking about these syllables as being the output of the brain to thinking about syllables as actually movements that animals generate to query the world around them. And with that shift in perspective, I think it's increasingly reasonable to think of syllables as being discrete things that are useful for the brain. And with that, I will end. I just want to mention again that the work that I've shown you today has been a collaboration between Bernard Sabatini, Scott Linderman, now Cheetah's lab, and my labs. The main people that have carried out the work that I've described to you today that's unpublished was Jeff Markowitz, who's a joint postdoc between me and Bernardo. He's now new faculty at Georgia Tech, along with Wynn Gillis and Maya Jay, who are some terrific graduate students. MoSeq was initially developed by Alex Wolchko and Matt Johnson. And there's a huge team of people who are listed here, including Tim and Mo, who are here at this meeting, who are continuing to work hard on and embellish motion sequencing. I thank you all so much for your attention, and I'm happy to take any questions.
Just a parenthesis, so as usual you have the microphones here, and I think there's someone walking around with microphones for questions. And on top of it, for people that, to stimulate people that are not so extrovert to participate too, you can go in the WOVA app, and you go in agenda, choose the session, choose the specific talk, and Q&A, and you can ask questions here, and Charlene will be checking that, so you can use that too.
Okay, there's a question down here in the front. Thank you to the folks running around with microphones.
Hello. Hi, thank you for the talk. I'm wondering if the embedding that MoSeq creates is interpretable without a downstream classifier. And for instance, can you tell that the nth feature corresponds to speed?
So can you just repeat the question? I can't hear you quite.
So I'm wondering if the embedding that MoSeq creates is interpretable without a downstream classifier. Like for instance, can you tell that the nth feature corresponds to speed? And then moreover, what do you think the role of interpretable embeddings is when combining machine learning with behavioral or neuroscience data?
Yeah, so obviously because we have an explicit generative model where our idea is that each one of these behavioral syllables is a kind of module of dynamics, we think that this is a very transparently interpretable kind of description of behavior, right? We're identifying these modules. The way that MoSeq identifies behavior, there's not like a clear axis, for example, on which velocity is described. Actually, Julia from Scott Linderman's lab, and I think I had up on my slide, has a poster on a version of MoSeq that we're developing that explicitly marginalizes out velocity to give you a kind of velocity knob for each syllable. So we've been thinking about that. I think in general, my thoughts are that, our goal, what is our goal here? Our goal is to try to come up with descriptions of behavior that give us insight into brain circuits and dynamics, and to try to understand how the architecture of behavior is created. And I suspect that the ethological insights that we kind of are leaning on reflected the interpretability of this organization of behavior. And so I think we favor models that are more rather than less interpretable.
Thank you.
I think there was a question over there.
Hello, may I ask a question? Hey Bob, this is Kay Tai from Salk and
Hey Kay, how are you?
Beautiful talk, really love your work and obviously super pioneering. My question is almost philosophical. So you have developed these tools to really get a quantitative grasp on behavior, and we are super inspired by all of that. But to come to the conclusion, for example, so traditionally in our field, we use behavior to sort of define whether a function has been perturbed or not. And so when you have the example of the dorsal striatal lesion where they no longer respond to TMT, you're like, oh, we know this mouse can still smell. But whether the impairment is at the detection, the perception, or the evaluation, ultimately the behavior is perturbed. So how do you know that the animal can still smell? I mean, obviously we have previous knowledge of what we think these structures do, but I'm just trying to get at the heart of how do you break down.
That's right.
Go ahead.
You're asking a bigger, I'm going to answer a small question, then I'll answer your bigger question. The answer to the small question is half my lab does olfaction, so we think about neural structures that respond to olfaction and can assess neural activity in response to TMT in the context of a DLS lesion. So that's how we know the animal can smell. The bigger question, that's the real question you're asking though is, we make a lesion, circuits are part of networks. How can you assert causality about anything? And I think here, I want to be very cautious in saying that I don't think, and I think many people in this audience would agree, that the striatum acts in isolation. I think it acts as part of a broad system in which there are associations that are made between context in the world and the potential availability of rewards and behaviors, right? And in that sense, I think what we've shown here is that there's at least one nidus there that seems to be important for a certain type of learning, but I don't want to extend my kind of claim about what DLS is doing much past that. Obviously, there's a whole circuit that's being recruited to define, for example, the behavioral state that the animal's in at any given moment, and how cues are going to get transformed into behaviors, and the DLS is one small part of that.
Let's alternate between the two sides, which have a diversity of cueing strategies. So let's go on over here.
How spatially structured are the spontaneous dopamine transients, and could transient in specific neurons be related to specific transitions?
Yeah, that's a great question. Alas, for both you and for me, I don't have the answer. We've just done photometry in dorsolateral striatum. The choice was actually governed by us wanting to duck underneath debates about whether the really relevant thing to look at is spiking activity in the midbrain versus kind of what's going on locally with dopamine axons. So instead, we just looked at the targets of that dopamine and have chosen to do photometry in this context. Obviously, as I'm sure you're aware, there's a huge diversity at the individual neuron level of dopaminergic responses to tasks and rewards and sensory cues. And we ourselves are very interested in what that diversity looks like in the context of spontaneous behavior. I just don't have any answers to your questions for now.
Hey Bob, this is Denise Kai from Mount Sinai. Great talk and congrats on tenure.
Oh, thank you.
So I have a question about the stability of the neural ensembles that is representing these behavioral syllables. And I thought it was super cool that you showed, these syllables, there's a repertoire, they exist, they're stable, and then depending on the environment or the situation, they would combine different syllables. So I'm curious whether these ensembles that represent each syllable also pre-exist and are stable. And so if the order of the sequence changes for the behavior, do the ensembles, are they stable, but then now they just change the order in which the ensembles play?
Yeah, so I can answer the latter question definitively, and then I'll duck underneath and babble about the first part. So if you have a given syllable, okay, and the order in which it's expressed changes, it's clear at the ensemble level that new neurons are dialed into the ensemble and some old neurons are dialed out, but there is a systematic relationship amongst these different ensembles such that we can use ensemble activity, to make accurate predictions about which syllable is being expressed, despite the fact that on any given trial, there's variability in the context in which that syllable is expressed, right? I think a different question, but it's a very important question that we don't have answers to yet is, if we were to do long-term recordings, how much drift would we expect to see in these neural representations for different kind of syllables and ensembles, and is there any kind of architecture that we can kind of understand about those neural ensembles? One result I didn't have time to tell you is that there seems to be essentially a systematic representation of behavioral space in dorsolateral striatum, whereas syllables that are similar have overlapping neural representations. So you can imagine that these could be stable across time, or you can imagine that there's a kind of representation that systematically rotates as time kind of proceeds forward. And actually Tim's here, he's very interested in setting up long-term kind of neurobehavioral recordings. I think it's one of the questions we'll end up addressing.
Thank you.
Hi. So really passionate presentation. You're putting the standard pretty high for the rest of
there you are. Thank you.
The meeting. I got two small questions, okay, which are pretty linked in my head. First, how do you select a number of syllables in your classification? And secondly, I guess that in everyday life, at some point, you realize you're doing like two stuff at the same time. Do you think there's some of those syllables where the animal are doing more stuff than in other syllables? Yeah, two different questions. The first is how do we decide how many syllables there are? So we don't. So we use a statistical method called the hierarchical Dirichlet process to learn from the data the number of syllables that affords us the ability to predict future behavior most accurately. And so this is the way that we kind of duck underneath the question of like how many syllables should we impose upon the model. We just let this method kind of learn this optimal kind of set of behavioral syllables. And I'll just mention that actually they're mathematically related methods that the ethologists themselves used 50 years ago that are related to this kind of discovery method. And the second question was about walking and chewing gum, right? So at any given moment in time, by definition, the animal's going to express one behavioral syllable, right? And so you might imagine that there could be compositions of behaviors where animals walk and chew gum, and you might wonder what MoSeq does if you're walking and chewing gum at the same time. So MoSeq, just by its nature, would identify a combination of behaviors like that as a new behavioral syllable. We're continuously thinking about ways to improve and reconceptualize MoSeq. And one thing that would be nice is if we had a kind of basis set that would not kind of always treat things that are kind of compositionally added as a new syllable. And we've been thinking with Scott a lot about that, and hopefully we'll make progress over time.
We're going to take the question from the app, followed by stage left.
So the question is by Samuel Liebana Garcia, and I'm just going to read it. It says, thanks for the great talk. What are your thoughts on the apparently conflicting observations that dopamine acts as a reinforcer for syllables, but at the same time increases variability in syllable transmissions?
Yeah, so there's a tension that for a lot of reasons I didn't have time to really unpack for you. And the tension is that on the one hand, I'm telling you that dopamine reinforces syllables, right, which should make behavior simpler. And at the same time, it's injecting variability, which should make behavior more complicated. So it's kind of doing both these things at once. So what gives with that? So we have some really interesting analysis that suggests that the balance between dopamine's ability to cause reinforcement and inject variability actually solves this problem where animals that happen to have a behavioral syllable that's associated with a high dopamine transient, you might imagine they would perseverate on that kind of associated syllable. They just do that behavior over and over again, and that would, of course, ruin the structure of naturalistic behavior. And so one of the things we're doing with these kind of fake reinforcement learning agents is, asking about the balance between dopamine's ability to inject variability and to promote reinforcement. And it's pretty clear that actually there's salutary effects in terms of maintaining the animal's ability to obtain reward without getting stuck by having both of these features co-encoded by dopamine.
Interesting talk. I had a question that built on two of the previous questions about the relationship between the neural dynamics and the behavioral syllabus dynamics. I guess I'm curious if you've tried to subject the neural ensemble data to the same sort of algorithm that you use on the behavioral data. And do you see syllabus alignment Or do you see alignment perhaps with the transitions between syllables? It seems like it would partly answer that question about.
It's such a wonderful question. It's an evergreen question. And actually, right, meaning that like one might imagine that you could use switching linear dynamical systems that are kind of like this MOSI system to characterize neural dynamics. Of course, Scott Linderman, who's here, has been a pioneer in that kind of space, both on behavioral data and neural data. We are actively building, actually, from what we think is interesting from our perspective, which are actually joint neurobehavioral models, where we incorporate both data streams to try to get more certainty over what a behavior is. And honestly, you know, at the end, I was kind of bloviating, right, about like what would be good for the future of you know, computational neuroethology. I think ultimately, building these joint neurobehavioral models is going to afford us a lot of certainty, and I think that's the perspective, at least we would like to adopt going forward.
Thank you.
I think we have time for one last question. Go ahead.
Am I the question? Is it me?
Go for it.
I have a totally unrelated question to what everyone else's questions is. You talked about the part of the talk where you were validating MOSI with these drug analyses as kind of just the segue to validate it, but I was wondering if actually that might be very informative about not necessarily the behavior, but the mechanism of the drugs. And I was wondering if you've done any analysis that tries to, like, I don't know, group the different drugs in terms of you're sort of laughing, so I assume you have, so I would love to know.
Yes. So I urge you to read our paper. We've done exactly the experiments that you've asked. So it turns out that we can kind of embed the MOSI-generated descriptions of drug-related behavior into a space. And the adjacencies in that space don't always reflect the characterized mechanistic targets of the drugs, but instead almost always reflect the known clinical effects of these drugs and their relationships, which is really, really cool. You know, most drugs that we're testing here are dirty drugs that hit a bunch of different receptors. We can also, based upon kind of dose-response curves, actually make predictions about which receptors are most impactful in terms of a given drug's overall behavioral response through this kind of system. And so actually we're quite interested in using the system to actually develop better psychopharmacology. That's a kind of side interest, but it is something that we're actively pursuing.
Wonderful. Thank you so much once again. To continue on the theme of brain and behavior, we have 3 contributed talks.
We can test if it
So these.
with the lights blinding me, can't really see me.
This is really intense, yeah.
Extend these displays.
There you go.
Yeah, can we put the pointer?
There you go. All right.
Okay.
Okay. So the first among the shorter talks is Atika Syeda. And after, I think the queuing up worked pretty well last time, so let's try that again after this talk as well. Okay, so anyone with questions, feel free to come down the aisle and queue up. Okay.
Hello, thank you. So today I will be presenting on interpretable behavioral features have conserved neural representations across mice. This is what neural activity looks like in a mouse visual cortex in complete darkness. But what's driving this activity in a sensory area? Several studies in rodents, fish, and flies have found that movements like running speed can modulate activity in sensory areas. More recent studies have observed that multiple dimensions of orofacial movements drive brain-wide neural activity. So to understand which of these behaviors are most important, I compared prediction of spontaneous neural activity in darkness by the different behaviors shown using a linear model. And I found that variance explained for test data by behaviors recorded from all these cameras was equivalent to variance explained by phase camera alone. So we now know that mouse facial behaviors are driving a large proportion of neural activity. We would like to understand the role of these behavioral representations in sensory areas. But to do so, we first need to track orofacial behaviors in an interpretable way. and study whether these behavioral representations are similar across mice. And are the behaviors interpretable? Previously, mouse face recordings were processed to obtain principal components of changes in mouse face represented as eigenfaces, which were used to predict neural activity. However, these principal components are not interpretable, as they fail to demonstrate complex behaviors such as whisking and sniffing. So to overcome the challenge posed by eigenfaces, I used another method for behavioral quantification, which is tracking key points. I labeled 15 distinct key points, which included key points for whiskers, mouth, nose, eye, and paw, hoping to capture various orofacial movements that mice may perform. Some of the current state-of-the-art tools used for tracking key points are Deep Lab Cut and Sleep that have different advantages. Deep Lab Cut uses ResNet to predict heat maps denoting probability for where a key point might lie, and simultaneously location refinement maps that contain for extracting the coordinates of key points in the full upsampled image. Sleep, on the other hand, employs a smaller network called UNet for predicting probability heat maps. So we wanted a model that's not only small and fast, but also accurate for tracking mouse orofacial movements. So we developed face map network that's smaller than Sleep and outputs probability heat maps and location refinement heat maps for better accuracy. Now for benchmarking, I compared our model with state-of-the-art tools by training all the models on the same training set that consisted of 2,400 frames obtained from 16 mice and recorded from different camera views. And I set aside 100 frames from a new mouse and camera angles as our test set. To compare the models, I computed inference speed of the models plotted on the x-axis. So the higher the better for tracking fast orofacial movements, and compared accuracy by computing test error in pixels on the y-axis. Sleep's default model had an inference speed of 200 frames per second, but it was less accurate than their slightly bigger model. Whereas Deep Lab Cut's default ResNet model had a better accuracy, but was slower than Sleep's default model. Deep Lab Cut's alternative model, MobileNet, had a faster inference speed, but decreased accuracy. Whereas our model, FaceMap Network, was not only as accurate as Deep Lab Cut's ResNet, but also 3 to 4 times faster. So it's easy to understand these improvements in speed, but does an improvement in accuracy of 2 to 3 pixels make a difference? So here is an example of test video prediction by Sleep and FaceMap. Sleep had some trouble tracking whiskers and mouth key points as they tend to jump from one region to another, but FaceMap consistently tracked the same set of whiskers. So FaceMap was able to maintain prediction accuracy while also improving on inference speed of Deep Lab Cut's most accurate model. Now that we have a good model for tracking mouse orofacial movements using key points, we come back to our question whether these key points can predict neural activity just as well as the eigenfaces shown earlier. For this, I obtained FaceMap key points and passed through a neural network that consisted of a linear layer and a set of convolutional filters, so to detect any oscillations and movement of key points from one facial region with respect to another facial region. The transform features were then passed through a bottleneck to get a reduced representation of behavior that was used to predict neural activity PCs. Now, comparing the variance explained for test data using eigenfaces and key points showed that key points model significantly improved our prediction of neural activity. To visualize this prediction of neural activity from behavior, here is a raster plot showing activity of 40,000 neurons over a few minutes. Neurons with similar activity patterns are put next to each other in the plot, and the running speed is shown at the top of the plot. We then replaced each row of the plot with the prediction of the neuron by the key points. You can see that orofacial behaviors tracked by key points can predict the global patterns of activity, and it also captures some of the finer features shared by a smaller group of neurons. So we now know that orofacial movements captured by key points can explain a larger proportion of neural activity. We would now like to study whether these behaviors are similar across mice. To do so, we first need to find a common behavioral framework for tracking facial movements for any mouse. So key points from a reference mouse were aligned with key points from a new mouse using affine transform to obtain key points in a shared coordinate space. Next, the multi-layer neural network described earlier was adapted to develop a multi-mouse model that was trained on key points from multiple different mice, such that all the weights of the core network were same across mice, except for the last layer, which was specific for a mouse. Now, comparing the variance explained for multi-mouse model with variance explained using eigenfaces and the single mouse model, I found that multi-mouse model predicted neural activity just as well as the single mouse model, suggesting that neural representations of behaviors are shared across mice. The multi-mouse model thus finds a lower dimensional representations of behaviors that are shared across mice. But are these shared behaviors interpretable? For this, latent features from the multi-mouse model were extracted and embedded into a lower dimensional space using t-SNE as shown here. So every point here represents a frame or time point in the video, and each point is colored by the running speed of the mouse at the time. You can see that some time points are grouped in certain regions of this low dimensional space. And by visualizing the frames in these cluster regions, you can identify certain behaviors, such as the resting phase when the mouse is inactive, and another region shows the mouse blinking. Another distinct region that was observed across mice was grooming. Whereas in the more active regions, you can see that the mouse was whisking while running, where its whiskers are pointing forwards. And another whisking pattern observed in active region was when the mouse moved its whiskers forward and back repeatedly. Hence, by using key points and the multi-mouse model, we were able to find a low-dimensional representation of behaviors that we can interpret. These behaviors are shared across mice and therefore may play a role in a canonical neural circuit. Finally, we have packaged our tool in FaceMaps GUI, which is available on GitHub, that can be used to track these facial key points on mouse face and explore low-dimensional features, thus providing a powerful tool for closed-loop behavioral experiments. Our goal is to have a model that's robust for any new mouse, and so we would like your contribution, and we're excited to use your mice videos to expand our training set for new camera views and different experimental rig setups, so that the model works out of the box for any lab. To summarize, I demonstrated FaceMap Network, which is a fast and accurate model for key points tracking of mouse orofacial movements, and the key points outperformed eigenfaces at predicting neural activity. The multi-mouse model showed that behavioral features are shared across mice and represented in a lower dimensional space. Ultimately, we hope to use FaceMap to understand how behavioral features integrated with sensory information, such as visual signals, can guide action or decision making. Finally, thanks to my colleagues Will and Renee for their contributions, and my supervisors, Marius and Carsen.
Thank you, Atika, for that beautiful talk and for facing such a large data set. Okay, so the mics are around for folks who have questions. I'll ask a quick one. You showed that video of the mouse grooming, and its paws was only, hands, paws, was coming up to the face only some of the times. How did you deal with that?
So this video where you mean like do you mean like when the key points are occluded?
Yeah, and how did, yeah.
Oh, right. So currently how how I label the data set is not, I didn't label the key points occluded, but when these raw predicted key points are filtered, meaning like if you apply median filter, then you can interpolate the values for these whiskers using where they might lie right before grooming and after. So currently, that's what the model is, but there are other ways how you could overcome like these occlusions, perhaps labeling those points during those time periods.
Thank you. This question over there.
Yeah, thank you for the great talk. Like really interesting tool and great results. I had a quick question about your latest comment on sharing videos and kind of extending the approach through FaceMap. What do you expect how robust your approach can be towards other types of video data that have different camera angles or different illumination factors, or like all the kind of variability that you expect to see across labs? Like, I think this affine alignment that you're doing is awesome. But I'm just wondering if I would ever be able to use it.
Right. So we have been trying to test with other labs generally and obtaining their videos, and mostly like some of the easier key points might be the eye key points and the nose key points. And then just depending on the resolution, how the videos were recorded, or sometimes adding different views, it might help the model be more robust. But most of the time, stable key points are tracked well. And just adding more data from different labs, and not a huge amount of data set, but few examples could help improve it much more.
Hi, quick question. I'm confused about a technical. I'm over here. Hi.
To the front on the right.
Okay. Hi.
Just a quick technical confusion on the model where you stitch the different mice data together. Don't you have to? So aren't those neural activity PCs going to be different on a per mice level? In which case, don't you need to have a rotation or affine back out to make that more useful?
Right. So that's exactly why our final layer is actually specific to the mouse. So once you're training on 4 different mice, each having different layers. But speaking of rotation, that's why we transform these key points before they're fed to the model, hoping to overcome that problem of different rotations. And that's something that's a work in progress in terms of achieving a good reference mouse and transformation so that it can be good represented for any representation of any new mouse.
Sure, thank you. You might try an extra rotation might make the latent space a little more reasonable. But yeah, thank you. Appreciate it.
Thanks.
Thank you very much.
Thanks.
Our next speaker is Renate Krause from the University of Zurich and ETH.
Okay, so hi everyone, and many thanks for this great opportunity. So in this talk today, I would like to introduce to you the concept of operative dimensions, and then show you how they are a very useful tool to get some insights into the usually very high dimensional connectivity matrices that you have in recurrent neural networks. But so more broadly speaking, we're studying this question of how is the network function related to the network structure. And we look at this in recurrent neural networks where the network function is usually given by these activities of the hidden units in these population activities. And the network structure is then stored in their connectivity stored in these weight matrices. And recent work has allowed us to gain valuable insights on different aspects of this question. And also in this work here, we're further studying how is the function related to the structure, but we specifically focus on recurrent neural networks which have been trained without any specific constraints or generated. And so what do I mean? To be more precise, so the networks that we look at are just vanilla recurrent neural networks, and we keep them quite small. They only have 100 hidden noisy units with a 10H activation function. And we then train these networks on 2 different tasks, so with gradient-based optimization. And the first one is the sine wave generation network, where the network receives an input, which can be of different levels, and then has to output a sine wave with the matching frequency. The second task is the context-dependent integration, where the network receives 2 sensory inputs, and then depending on the context input, it has to integrate this input over the course of the trial. But so now the exact details of these tasks are not very important, but what recent work has showed very consistently is that the network function of these networks is usually low dimensional. So this means if you look at the principal components of the network activity and how much variance that they explain, we see clearly that the first few principal components explain most of the variance, meaning this network activity lives in a low dimensional subspace. And we can now ask the same question for the weight matrix. And in this talk, I will focus on the column dimensions of the weight matrix, but in general, the results that I'm showing you are very, very similar for the row dimensions. So if we now perform PCA on the weight matrix, what we see is that it looks very different. So we see that the variance explained for the different principal components decays only very, very slowly, indicating that this weight matrix is actually high dimensional. And so to make this a bit more intuitive or better understandable, if we look at these weight matrices, we can understand one column of it as all the output connections of a single unit. So this means we can understand one column vector as a direction in state space, which tells you in which direction that this unit is sending its output. So this red unit would point in this direction, the yellow in this direction, and so on. And so now the fact that this weight matrix is high dimensional actually tells us that this weight matrix, or the units, here can send their outputs in all kinds of directions roughly equally. And so now, together, this is kind of puzzling, because it says your activity lives in a low-dimensional subspace, and yet the underlying weight matrix, which actually generates all these activities, says all these units can send their outputs in all kinds of directions. And so this naturally raises the question, well, do we really need all of these dimensions then? And so to look at this question, we did the following analysis. So this is very, very strongly inspired by previous work on the low-rank recurrent neural network framework by Francesca Mastrogiuseppe and Friedrich Schüssler. And the analysis that we're doing is similar to like a lesion study. So we just take the weight matrix and then we sequentially remove these principal components, these high variance dimensions, and then just ask, is the network still able to perform the task? And so this means now at this end of the plot, we would run the full rank network, so the original network. And then as you go to the left, we start to remove dimensions, these principal components from the weight matrix, and then look at the output error of the network. And so when we now look at this plot, we can make 2 main observations. So the first one is that it doesn't really match what we saw in the network function, because we need more than 75 out of 100 dimensions to get back to the original performance. And the second observation is that we have these very sudden jumps at specific ranks. And this is odd, given that we've seen all the principal components actually explain a very similar amount of the total variance. And so now, having this in mind, there are kind of 2 possible scenarios how to understand this. The first one is that maybe it is just truly high dimensional, and then we have to go ahead and further understand what these dimensions are doing. But the second option is that maybe these principal components, or high variance dimensions, they are just not well aligned with any functionally relevant dimensions. And that is why we need so many of them. And so we decided then to kind of distinguish between these 2 scenarios, that we would like to define a new type of dimension. And we want to define these dimensions such that they're much more closely related now to the actual network function. And this is what we then refer to as these operative dimensions. And so how can we find them or define them? So we want to define these operative dimensions, we want to move away from this very global way of looking at the network function, which is just the performance, and instead look at the local dynamics. So specifically, we define these operative dimensions as those dimensions which, if you remove them from the weight matrix, have the largest impact on these local dynamics. And so how can we quantify a local impact? So let's say you're just at a random location in your state space, just one location. And then what we do is we just run the network for 1 time step to collect the local recurrent flow field. So you see here, this is just the normal RNN equation, the original full rank network, but without input and without noise. So we place the network in X of t, we run it for one time step, collect the endpoint. And now we just do exactly the same, but with our reduced rank network to collect these reduced rank dynamics. So you take the weight matrix, you move it arbitrary dimension, but then you again just run the network for one time point and collect the endpoint. And now to define what is the impact, we simply measure the Euclidean distance between these two endpoints. So the larger the distance, the larger the impact. And now that we have this very quantifiable measure to define impact, we can just use this to optimize over it to actually find now our operative dimensions. And so we do this in 3 steps. So, and in the first step, we define a number of sampling locations, which help us define which part of the dynamics that we're actually interested in. And because we are interested in the functionally relevant dynamics, we place them on these condition average trajectories. So just to make this very clear, here as an example for the sine wave generator network, we can get now one condition average trajectory for every input level, so we average over the noise and different trials. And then here you again see the different condition average trajectories, and the white dots indicate the sampling locations, and we just place them equally spaced in time along these trajectories. And so now let's focus on just one of them for now. So here again in white, the different sampling locations equally spaced in time. Because now what we do is we just run this optimization. So we ask if you are at one specific location, which dimension would you move from your weight matrix so that you can maximize the change in your local dynamics. So you maximize the impact. And we can do this once to get the first operative dimension and just keep on repeating that to get the second and third operative dimension and so on. And then similar to PCA, we go ahead and say, okay, these next upcoming dimensions have to be orthogonal to all the previously identified ones. And with this step, we can now collect one set of local operative dimensions at every single of our sampling locations. So not just this mean trajectory, but all the sampling locations. And once we have this, we can just collect all of them. So they all live in the same space. We just scale them by their local impact and then perform PCA on this set of dimensions to get then one set of global operative dimensions per network. And now that we have our new type of dimensions, we can go back to the previous analysis and again ask how many dimensions does it really need. And now this plot looks very different. So now we see here it's exactly the same plot. So we look at the output error, but now instead of removing the principal components, we remove the operative dimensions that we just defined. And now we see that the network actually only needs around 15 out of 100 of these dimensions to perform the task with the original performance. And this clearly indicates that actually a low-dimensional subspace in this originally high-dimensional weight matrix is sufficient to achieve the original performance. And this plot now also looks very similar to what you've seen previously for the dimensionality of the network function. And we can also show more generally that actually the number of operative dimensions that a network would need to perform the task is limited by the number of dimensions that you have in your network activity. So low-dimensional activity means low-dimensional subspace in your weight matrix. And just as a control, we also then went ahead and collected these local operative dimensions that just random sampling locations in state space instead of along these condition trajectories. And this here just to show that it isn't completely trivial to identify the subspace. And so now all this together kind of indicates really that actually a large part or large subspace of this weight matrix seems functionally irrelevant. But just to also point this out, it also raises the question then, well, which RNNs do we want to study and what would we expect in biology? And with this, I would like to conclude. So we started out by asking how is the network function related to the network structure. We then studied this in unconstrained recurrent neural networks, which have typically low-dimensional dynamics and high-dimensional connectivities. And we then went ahead and defined these new type of dimensions, these operative dimensions, to describe the functionally relevant subspace in the network connectivity. And we've seen then that this is actually low-dimensional is enough to achieve the original performance. And with this, I again like to point out that this was worked together with Matthew Cook, Valerio, Mante, and Giacomo Indiveri, and many nice colleagues in the Institute of Neuroinformatics in Zurich. Thank you very much for your time.
Thank you, Renate. That was a great talk connecting function and dynamics. It looks like there's a few people lined up already over there to ask you questions.
Hi, thanks for a great talk. Quick question. Was the initial W before training non-zero? And if so, did you look at the difference Did you try to look at the PCs of the delta W?
What do you mean the initial data? You mean the initial weight matrix?
You trained the network, so before training you initialize W with some random connectivity.
Yeah.
And then it's possible to take the final connectivity and subtract the initial one. And I wonder whether you had a look at that.
We have not looked at that, but I can tell you that also the initial network was already high dimensional, and generally the dimensionality of the weight matrix barely changed across training. If that answers your question, partially.
Hey, over on your right side at the front.
Thanks.
Really beautiful talk. Maybe I missed exactly how the optimization works, but I'm wondering how much of it, how close does the output look like what you'd get if you were looking at the SVD of just like the input dimensions? Like, is the nonlinearity critical to this, where you're following the actual trajectories, or is it related to, you know, the inputs of the matrix?
So you're asking if the dimensions that we find with this optimization are similar to the principal components? Is that what you're asking?
To the, what is it, left singular vectors?
Yeah,
the input
exactly.
Side?
So the first few, I can show you. All right, just answer. So the first few are not similar, but interestingly, they are very similar to the principal components of the network activities. But for the nonlinearity, so for the column dimensions, you would want to look at the activity before the nonlinearity, and for the row dimensions after the nonlinearity. So in that sense, the nonlinearity is a key part to then find the right ones.
Let's take one more question.
Hi.
Hi.
So you showed that the principal components of the weight matrix don't really tell you much about these behaviors. I was wondering if you try to look at like the controllability and the observability Gramian instead, which, at least for linear dynamics, are going to be more informative as to the kind of, you know, impact a particular direction will have on the dynamics of the system.
So I didn't understand the...
Observability and controllability Gramians, and the eigenvectors of those.
I'm not sure I understand the question. I'm very sorry. Wait, maybe I can talk to you later.
It's probably, yeah, for linear dynamics, it's probably useful to look at those matrices because they are, you know, going to be more informative as to what happens in the dynamics of the system, especially when it's
So I mean, one thing, we looked at the local linear dynamics. Is this what you're asking? And then we can say if we remove the dimensions, the local linear dynamics, they do not change. The dominant local dynamics do not change, but the other ones do.
Okay. Thank you, Raj, once again.
Thank you.
Just a quick comment on the last question. I don't know who asked, but I think a notion of nonlinear observability and controllability Gramians do exist, but they get pretty hairy pretty fast. Okay. And now I have the pleasure of introducing Elliott Abe from the University of Oregon.
Hello, everyone. Like being said, my name is Elliott Abe, and I'm a PhD student at the University of Oregon Institute of Neuroscience, co-advised by Dr. Chris Niell and Dr. James Murray. Today, I'm going to talk to you about some of my recent work that has come out of the Niell lab related to how visual information and eye and head position integrate within V1 of freely moving mice. So traditionally, visual neuroscience has been conducted under head fixation, where the experimenter has precise control over what the mice see, and then can be later correlated with neural activity. While these experiments have revealed a lot about how the visual system works, they did not address the fact that under natural conditions, eye, head, and body movements create a very different and more complex visual scene, shown on the bottom video here. Additionally, from previous work, we know that neurons in V1 are not simple feature detectors. For example, in this plot here, we know that orientation tuning curves are modulated by locomotion in head fixed conditions. And in unrestrained conditions, the time around head orienting movements is modulated by vertical head movements. So given the challenge of unrestrained behavior, like not having the control over what the mice see, we set out to kind of tackle 3 topics. First was to develop a method to investigate visual coding in freely moving mice. Next was to see if and how does eye and head position modulate neural activity. And ultimately, we wanted to find out how does eye and head position integrate with visual input during free movement. So for this first part, to develop a method, there are a couple of things that we needed. First, we needed to know what the mice are seeing and where the mice are seeing, looking in that visual scene. Additionally, we needed the neural activity simultaneously during this time, and we needed to figure out a way to relate the retinal input to the neural activity. So the following experimental work was led by Phil Parker, who is an outstanding postdoc in the Niell lab. And to address the challenge of no longer having control over what the mice see, we developed a miniature head-mounted camera system. And within this system, there is a world camera on top of the mouse's head that records the visual scene from the mouse's perspective. A camera pointed at the eye so we can know where in the visual scene the mouse is looking. A silicon probe in V1 to record neural activity. And finally, an inertial measurement unit to record the pitch and roll of the head. So altogether, this is what the data looks like. And on the top, we have the visual scene from the mouse's head perspective, the eye, the horizontal eye position, the vertical pitch of the head. And on the bottom here is over 100 simultaneously recorded units while the mice is freely moving. And so what we can see is that when the mouse is stationary, the eye is stable and so is the visual scene, but once the mouse starts to move, there's a drastic shift in neural activity. With this data, it gets us part of the way to doing freely moving visual physiology. However, the world camera here is in a head-centric perspective. And in order to relate this to the neural activity, we need to transform this into a retinocentric perspective. So to do this, we used a shifter network that took as input the horizontal eye position, the vertical eye position, and the pitch of the head as a proxy for cyclotorsion of the eye. We then outputted the parameters of horizontal shift dx, vertical shift dy, and a rotation of the image d alpha. We then shifted the image and then fed this into a GLM network that then predicted single unit activity. This entire system was then trained end-to-end such that the transformation parameters that were found were the ones that best predict the neural activity population overall. So on the bottom here is a video of what this correction looks like, where on the left side is the raw head-centric point of view, and on the right here is the corrected retinocentric one. With this, we can now have a model to be able to investigate how visual information is encoded during free movement. So using the now approximate retinal input, we can train the GLM network to predict single units. And the weights of the GLM here are going to represent the spatial receptive fields of these individual units. And what we find is, and this is 4 example units here, is these receptive fields match our intuition of receptive fields mapped through traditional means with on and off subregions that are spatially localized and oriented. And to our knowledge, these are the first visual receptive fields mapped during free movement. With this, we know where in space the neurons are responding to, but how well do these receptive fields predict the neural activity at a moment-to-moment basis? So in this plot here, in black, is the firing rate over time of Unit 3. And in green here is our prediction of the model. And as you can see, overall, the prediction is pretty good with a correlation coefficient of 0.4. And across the population, on average, the correlation is around 0.3. Additionally, as a sanity check, we wanted to know how does the freely moving receptive fields compare with those receptive fields measured using traditional methods. To this end, during the experiments, the receptive fields were first mapped in a head-fixed condition with white noise stimulus and then allowed to freely roam around. Then we can look at the receptive fields of the same neurons across these 2 conditions and see how well they match. And what we found was pretty amazing, that with the correction from the shifter network, we get the same receptive fields between 2 completely different conditions. And these are the 4 example units, where the correlation coefficient at the top here is the pixel-wise correlation between the receptive fields at each location. And we find that the receptive fields are matching in both spatial location and the number of subunits. So across the population, what we find is that 30% of the units had a significant matching receptive field shown in black as compared to a shuffle distribution in gray. So for our first part, we were able to successfully map receptive fields during free movement that allows us to do visual physiology. with unrestrained behavior. Now we want to look to see if and how eye and head position modulate neural activity in V1. And what we find was pretty surprising is that, yes, we do see that there's significant modulation of the neural activity. So these are two example tuning curves for the horizontal eye position and the pitch of the head. And we see that there is positive modulation based off of the angle of the eye and the head. With these tuning curves across the population, we can calculate a modulation index. And across the population, we find there's a diversity of modulation, with 25% of neurons having a modulation index above 0.33, meaning a doubling of firing rate. With this, we can now train a position-only GLM, where we give the input only the eye and head position to then predict the neural activity. And we find that this is able to predict the neural activity fairly well, but not quite as well as the visual-only model shown in green. So there was about a 0.3 correlation with the position-only and a 0.45 with the visual-only model. And across the population of neurons, where each dot here represents a neuron, we find on average that the vision-only model does a better job at predicting the neural activity. So now we were able to answer this question and found that there is tuning to eye and head position. And finally, we can get to the last part, which is how does eye and head position integrate with visual movement, input during free movement? So there are two possible hypotheses for the modulation of input, either linearly, where these two signals can vary independently and then sum, or through a nonlinear multiplicative gain modulation. To test this and figure out which one, we took the trained visual network, froze the weights so that the additive and multiplicative models would have the same visual information. We then added a positional network that took the eye and head position as input. The output of these two models were then either added or multiplied together to predict single unit activity. And this is what we find. So as a reminder, this is our example unit for the visual only and the position only networks. We then take the visual input and the position input and see how well does the multiplicative model do. And we find that there's a significant increase in the ability to predict neural activity. And across the population, we find there's a systematic increase when incorporating eye and head position into our model, where each dot here represents a neuron, and the color of the dot is represented by which model had a higher correlation value. Now, to compare additive and multiplicative models, we took the difference between the correlation values and found that most neurons had a higher correlation value for the multiplicative model, which is pretty exciting because the study of multiplicative interactions or gain fields in V1 has a lot of theoretical history. And one of these functions is that it allows for coordinate transformations of representations. So for example, from a venotopic perspective to a head-centric to then a allocentric representation. To conclude, we made a method to perform freely moving visual physiology. We measured the first freely moving visual receptor fields. We found positional tuning for eye and head position in V1 neurons. And finally, we found that visual input and eye and head position predominantly integrate through a multiplicative gain interaction. Taken together, this work opens up the possibility to study visual coding during natural behavior. Thank you, everyone, for listening, and thank you for the Niell and Murray Labs for the support. If you want to learn more about this work, we have a bioRxiv paper at this QR code, and all of the code is freely available at this GitHub link QR code. I will take any questions.
Thank you, Elliott. And yet another very impressive and courageous set of experiments. Let's take the first question over there.
I noticed that when you showed the model versus the data, then there were lots of times where it fit very well, and then short epochs where the fit wasn't that good, or let's say was the worst. Did you have a look at what happened during these times when the error was maximal?
We haven't quite looked exactly what's maximum yet. One thing is that this is also a linear model. So all the interactions of when it's really well fit could be just linear operations of those two parameters, but we haven't then accounted for, say, like other modulatory signals that could happen in a non-linear way.
Can I ask another one here?
Sure.
What question over here?
Oh, hello. So I had a question. Have you been able to dissociate the effects of sort of absolute eye and head position versus rates of change of those?
Yeah.
Is one more effective than the other?
Great question. Yeah. So all of this is in terms of absolute position. What is the position of the eye and the head? We are currently looking into what is the rate of change part. It's a little bit more complicated because those variables are more dynamic while these are static. And you know, that's definitely in the works, and we have some analyses lined up for that.
Hey, Elliott, really cool stuff. I just wanted to ask, so you showed us some data on spontaneous behaviors and sort of stabilization of gaze relative to the head. I've seen before some of your work and Phil's work showing some really cool bobbing of the head behavior and jumping behavior. So I'm wondering if you could tell us anything at all about how a sort of motivated behavior might affect V1 activity too.
Yeah, great question. Currently, we haven't applied this system to any of the motivated behavior, but that is the future plan. The movements of the eyes are often highly correlated with movements of the head. So if, for example, in the jumping behavior, when you're moving your head, your eyes are going to be compensating for the visual field. And so we expect something similar to be able to happen there. The idea of correcting the head-centric visual field was to get an approximate retinal image. And regardless of kind of how the mouse is moving its head or eye, that's kind of the ultimate goal, is so that can we get a approximate retinal input.
Very cool, thank you.
Okay, thank you, Elliott, once again.
Yep.
The talks may be over, but the fun is just beginning. Those of you who are jet lagged and can't sleep anyway, go to the poster session.